Welcome to our exploration of data collection techniques in biological research. In the world of biology, gathering accurate and reliable data is the foundation of every discovery. Whether scientists are studying the behavior of animals, the growth of plants, or the genetics of microorganisms, the methods they use to collect data can make all the difference. Today we'll journey through the essential techniques that researchers rely on, highlighting their importance and how they shape our understanding of life. Let's begin by understanding why data collection is so crucial in biological research and what makes it unique compared to other scientific fields. Data is the backbone of biological research. Without it, scientists would be left with only theories and assumptions. Accurate data allows researchers to test hypotheses, draw meaningful conclusions, and build upon previous work. In biology, the complexity of living systems means that data must be collected carefully and systematically. This ensures that results are reliable and can be replicated by others. From field studies to laboratory experiments, the quality of data collection directly impacts the credibility of research findings. Now, let's look at the main categories of data collection techniques used in biology. One of the oldest and most fundamental data collection methods in biology is observation. Observational techniques involve watching and recording the behavior or characteristics of organisms in their natural environment or under controlled conditions. This method is especially valuable for studying animals in the wild, plant growth, or microbial activity. Observations can be qualitative, such as noting color changes, or quantitative, like counting the number of individuals in a population. The key is to remain objective and minimize interference with the subjects being studied. Let's explore how these techniques are applied in real research scenarios. Field studies are a cornerstone of biological research, allowing scientists to collect data directly from natural habitats. Researchers venture into forests, oceans, grasslands, and other ecosystems to observe and record information about organisms and their interactions. This approach provides valuable insights into how species adapt to their environments and respond to changes. Field studies often require careful planning, specialized equipment, and a keen eye for detail. The data gathered can include everything from animal migration patterns to soil composition. Next, we'll see how sampling methods are used to make sense of large populations. Sampling is essential when it's impossible to study every individual in a population. Biologists use various sampling methods to select representative groups for study. Random sampling ensures that every member has an equal chance of being chosen, reducing bias. Systematic sampling involves selecting individuals at regular intervals, while stratified sampling divides the population into subgroups and samples from each. These techniques help researchers draw accurate conclusions about entire populations based on a manageable subset. Let's dive deeper into how random sampling works in practice. Random sampling is a powerful tool in biological research. By giving every individual in a population an equal chance of selection, researchers can avoid unintentional bias. This technique is often used in ecological studies, such as estimating the number of fish in a lake or the diversity of plants in a meadow. Tools like random number generators or grid systems help ensure true randomness. The resulting data is more likely to reflect the true characteristics of the population. Next, let's look at systematic sampling and how it differs from random sampling. Systematic sampling involves selecting individuals at regular intervals from a list or a physical space. For example, a researcher might sample every 10th plant along a transect line in a forest. This method is straightforward and easy to implement, especially when dealing with large populations. However, it's important to ensure that the interval doesn't coincide with any underlying patterns in the population, which could introduce bias. Systematic sampling is commonly used in vegetation surveys and animal population studies. Now, let's see how stratified sampling helps researchers study diverse populations. Stratified sampling is used when a population has distinct subgroups, or strata, that need to be represented. Researchers divide the population based on characteristics like age, habitat, or species, and then sample from each subgroup. This ensures that all relevant groups are included in the study, leading to more accurate and meaningful results. Stratified sampling is particularly useful in studies of biodiversity, where different species or habitats must be considered. With a solid understanding of sampling methods, let's move on to experimental techniques in biological research. Experimental techniques are at the heart of many biological discoveries. In controlled experiments, researchers manipulate one or more variables to observe their effects on a biological system. This approach allows scientists to establish cause and effect relationships, which are essential for understanding complex processes. Experiments can be conducted in laboratories or in the field, depending on the research question. Key elements include control groups, randomization, and replication to ensure reliable results. Let's take a closer look at how control groups are used in biological experiments. Control group is a fundamental part of any well-designed experiment. It serves as a baseline for comparison, allowing researchers to determine the effect of the variable being tested. For example, in a study on plant growth, one group of plants might receive fertilizer while the control group does not. By comparing the two groups, scientists can isolate the impact of the fertilizer. Control groups help eliminate alternative explanations and strengthen the validity of the result. Next, we'll discuss the importance of randomization in experiments. Randomization is a key principle in experimental design. By randomly assigning subjects to different groups, researchers minimize the risk of bias and ensure that differences between groups are due to the experimental variable, not other factors. This technique is widely used in clinical trials, genetic studies, and ecological experiments. Randomization increases the reliability of results and makes findings more generalizable. With control groups and randomization in place, let's look at the role of replication in biological experiments. Replication means repeating an experiment or observation to confirm the results. In biology, replication is crucial for ensuring that findings are not due to chance or unique circumstances. By conducting multiple trials or using several samples, researchers can assess the consistency of their results. Replication also allows others to verify findings by repeating the study under similar conditions. This strengthens the credibility of scientific knowledge. Now, let's explore how surveys and questionnaires are used to collect data in biological research. Surveys and questionnaires are valuable tools for collecting data from large groups of people. In biological research, they are often used to gather information about human behavior, health, or environmental practices. Well-designed surveys can provide insights into patterns and trends that would be difficult to observe directly. Researchers must carefully craft questions to avoid bias and ensure clarity. The data collected can be analyzed statistically to identify correlations and inform further research. Next, we'll look at the use of interviews in biological studies. Interviews allow researchers to collect detailed qualitative data from individuals or groups. In biological research, interviews might be used to understand local ecological knowledge, health behaviors, or conservation attitudes. This technique provides depth and context that surveys alone may miss. Interviews can be structured with set questions or unstructured, allowing for open-ended discussion. The information gathered can help shape research questions and interpret findings. Now, let's discuss the importance of laboratory techniques in data collection. Laboratory techniques are essential for collecting precise and controlled data in biological research. These methods include everything from microscopy and DNA analysis to chemical assays and cell culture. Laboratories provide a controlled environment where variables can be carefully managed, allowing for accurate measurement and repeatability. Advanced equipment and standardized protocols ensure that data is reliable and comparable across studies. Let's take a closer look at microscopy, one of the most widely used laboratory techniques in biology. Microscopy allows scientists to observe structures that are too small to be seen with the naked eye. From cells and bacteria to tiny organelles, microscopes reveal the hidden world of biology. There are various types of microscopes, including light, electron, and fluorescence microscopes, each suited to different research needs. Microscopy is fundamental in fields like cell biology, microbiology, and histology. The images and measurements obtained provide critical data for understanding biological processes. Next, we'll explore DNA analysis as a data collection technique. DNA analysis has revolutionized biological research. Techniques like PCR, sequencing, and electrophoresis allow scientists to study genetic material in detail. DNA data can reveal relationships between species, track disease outbreaks, and identify genetic mutations. These methods require specialized equipment and careful sample handling to prevent contamination. The information gained from DNA analysis is invaluable for fields such as genetics, evolutionary biology, and medicine. Now, let's look at chemical assays and their role in data collection. Chemical assays are used to measure the concentration of substances in biological samples. For example, researchers might use assays to determine the amount of glucose in blood or the presence of toxins in water. These techniques rely on chemical reactions that produce measurable signals, such as color changes or light emission. Chemical assays are widely used in physiology, environmental science, and clinical diagnostics. The data collected helps scientists understand biological functions and detect abnormalities. Next, we'll discuss the use of cell culture in research. Cell culture involves growing cells in controlled laboratory conditions. This technique allows researchers to study cell behavior, test drug effects, and investigate disease mechanisms. By providing the right nutrients and environment, scientists can observe how cells grow, divide, and respond to stimuli. Cell culture is essential in cancer research, vaccine development, and regenerative medicine. The data collected from cell cultures provides insights that are difficult to obtain from whole organisms. Let's now explore the importance of data recording and management. Accurate data recording is vital in biological research. Researchers use notebooks, spreadsheets, and specialized software to document observations, measurements, and experimental results. Consistent and detailed records ensure that data can be analyzed, shared, and reproduced by others. Proper data management also helps prevent errors and loss of information. As research becomes more complex, digital tools and databases play an increasingly important role in organizing and storing data. Next, we'll look at the use of sensors and automated data collection. Modern biological research often relies on sensors and automated systems to collect data. These tools can monitor environmental conditions, track animal movements, or measure physiological parameters in real time. Automation reduces human error and allows for continuous data collection over long periods. Examples include weather stations, GPS collars, and automated blood analyzers. The data generated is often large and complex, requiring advanced software for analysis. Let's now discuss the importance of ethical considerations in data collection. Ethics play a crucial role in biological data collection. Researchers must ensure that their methods respect the welfare of animals, the privacy of human subjects, and the integrity of ecosystems. Ethical guidelines require informed consent, humane treatment, and minimal environmental impact. Institutional review boards and regulatory agencies oversee research to ensure compliance. Ethical data collection not only protects subjects but also enhances the credibility and acceptance of research findings. Next, we'll look at the importance of pilot studies in refining data collection techniques. Pilot studies are small-scale preliminary studies conducted before the main research project. They help researchers test and refine data collection methods, identify potential problems, and estimate the resources needed. By conducting a pilot study, scientists can improve the design of their experiments and increase the likelihood of success. Pilot studies are especially valuable when using new techniques or working in unfamiliar environments. The feedback gained helps ensure that the main study runs smoothly and produces reliable data. Now, let's explore the use of remote sensing in biological research. Remote sensing involves collecting data about the environment from a distance, often using satellites or drones. This technique is widely used in ecology, agriculture, and environmental monitoring. Remote sensing can provide information on land use, vegetation cover, and changes in ecosystems over time. The data collected is often processed using Geographic Information Systems (GIS) to create detailed maps and models. Remote sensing allows researchers to study large areas that would be difficult or impossible to access directly. Next, we'll discuss the role of citizen science in data collection. Citizen science involves members of the public in data collection and research. Volunteers might count birds, monitor water quality, or record weather observations. This approach expands the reach of scientific studies and engages communities in the research process. Data collected by citizen scientists can be valuable for tracking trends and identifying changes in the environment. However, researchers must provide clear instructions and quality control to ensure data reliability. Now, let's look at the use of archival data in biological research. Archival data refers to information collected in the past and stored in databases, libraries, or museums. Biologists often use archival data to study long-term trends, compare historical and current conditions, or analyze rare events. Examples include weather records, museum specimens, and genetic databases. Using archival data can save time and resources, but researchers must consider the quality and relevance of the information. Next, we'll discuss the importance of calibration and standardization in data collection. Calibration and standardization ensure that data collected from different sources or instruments are comparable. Calibration involves adjusting equipment to known standards, while standardization means using consistent methods and units. These practices are essential for producing reliable and reproducible results, especially in collaborative or long-term studies. Without proper calibration and standardization, data may be inaccurate or misleading. Now, let's look at the use of photographic and video documentation in biological research. Photographic and video documentation are powerful tools for recording observations and experiments. Images and videos provide visual evidence that can be analyzed, shared, and archived for future reference. In biological research, photographs might capture animal behavior, plant growth, or microscopic structures. High-quality documentation supports data accuracy and allows for detailed analysis. With advances in digital imaging, researchers can now collect and process large volumes of visual data efficiently. Next, we'll discuss the use of modeling and simulation in data collection. Modeling and simulation are increasingly important in biological research. By creating mathematical or computer models, scientists can simulate biological processes and predict outcomes. These models are built using data collected from experiments, observations, and literature. Simulation allows researchers to test hypotheses, explore scenarios, and identify patterns that may not be visible in raw data. Modeling is especially useful for studying complex systems such as ecosystems or disease spread. Now, let's look at the importance of data validation and quality control. Data validation is the process of checking data for accuracy, completeness, and consistency. In biological research, this step is essential to ensure that conclusions are based on reliable information. Validation may involve cross-checking data with other sources, running statistical tests, or reviewing records for errors. Quality control procedures help identify and correct mistakes before analysis. By validating data, researchers increase the credibility and impact of their finding. Next, we'll discuss the importance of metadata in data collection. Metadata is data about data. It provides context such as how, when, and where data was collected, what methods were used, and who collected it. In biological research, metadata is crucial for interpreting results, sharing data, and ensuring reproducibility. Without proper metadata, valuable information can be lost or misunderstood. Researchers are encouraged to document metadata alongside their primary data, using standardized formats when possible. Now, let's look at the role of big data and bioinformatics in modern biology. Rise of big data has transformed biological research. With advances in technology, scientists can now collect and analyze massive datasets, from genome sequences to environmental sensor readings. Big data enables researchers to identify patterns, make predictions, and uncover relationships that would be impossible to detect with smaller datasets. Bioinformatics, the use of computational tools to manage and analyze biological data, is a rapidly growing field. Let's explore how bioinformatics supports data collection and analysis. Bioinformatics combines biology, computer science, and statistics to analyze complex biological data. Tools like sequence alignment software, databases, and visualization platforms help researchers manage and interpret large datasets. Bioinformatics is essential for genomics, proteomics, and systems biology. These tools automate data processing, reduce errors, and enable new types of analysis. As data collection becomes more sophisticated, bioinformatics will continue to play a central role in biological research. Next, we'll discuss the importance of collaboration in data collection. Collaboration is increasingly important in biological data collection. Large-scale projects often require teams of scientists with diverse expertise, as well as partnerships with institutions and communities. Collaborative efforts allow for the sharing of resources, data, and ideas, leading to more comprehensive and impactful research. International collaborations can address global challenges, such as disease outbreaks or biodiversity loss. Effective communication and data sharing are key to successful collaboration. Now, let's look at the challenges faced in biological data collection. Collecting data in biological research is not without challenges. Researchers may face difficulties such as limited access to study sites, unpredictable weather, equipment failures, or ethical dilemmas. Data quality can be affected by human error, sample contamination, or incomplete records. Addressing these challenges requires careful planning, flexibility, and adherence to best practices. By anticipating potential problems, scientists can develop strategies to minimize their impact. Next, we'll discuss the importance of continuous learning and adaptation in research. Biological research is a dynamic field, and data collection techniques are constantly evolving. Researchers must stay informed about new methods, technologies, and best practices. Continuous learning ensures that data collection remains accurate, efficient, and relevant. Attending workshops, reading scientific literature, and collaborating with peers are all ways to keep skills up to date. Adaptability is key as new challenges and opportunities arise. Now, let's look at the role of open data and data sharing in advancing biological research. Open data initiatives encourage researchers to share their data with the broader scientific community. By making data publicly available, scientists promote transparency, reproducibility, and collaboration. Open data can accelerate discoveries, avoid duplication of effort, and enable new analyses. However, sharing data also requires careful attention to privacy, intellectual property, and data quality. As open data becomes more common, researchers must balance openness with responsibility. Next, we'll discuss the future trends in biological data collection. Future of data collection in biological research is bright and full of innovation. Emerging technologies like artificial intelligence, machine learning, and advanced sensors are transforming how data is gathered and analyzed. These tools can process vast amounts of information quickly and uncover patterns that were previously hidden. As technology advances, researchers will be able to collect more detailed and accurate data, leading to new discoveries and deeper understanding. Let's now summarize the key points from our journey through data collection techniques. Throughout this video, we've explored a wide range of data collection techniques used in biological research. From observation and sampling to laboratory experiments and remote sensing, each method has its strengths and applications. Accurate data collection is essential for testing hypotheses, understanding biological processes, and making informed decisions. By combining traditional and modern techniques, researchers can tackle complex questions and contribute to scientific progress. Next, let's reflect on the impact of data collection on society and the environment. Data collected in biological research has far-reaching impacts on society. It informs public health policies, conservation efforts, and environmental management. Reliable data helps policymakers make decisions that protect human health, preserve biodiversity, and ensure sustainable use of resources. By sharing their findings, biologists contribute to education and public awareness. The benefits of accurate data collection extend beyond the laboratory, shaping the world we live in. Now, let's consider the ethical responsibility that comes with collecting and using biological data. With great power comes great responsibility. Biologists must use data collection techniques ethically, respecting the rights of individuals, communities, and ecosystems. This includes obtaining informed consent, protecting sensitive information, and minimizing harm. Ethical research builds trust between scientists and the public, ensuring that discoveries benefit society as a whole. As data collection methods evolve, maintaining high ethical standards remains essential. Next, we'll highlight the importance of communication and transparency in research. Effective communication is vital for sharing research findings and data collection methods. Scientists must present their data clearly and accurately, using language that is accessible to both experts and the public. Transparent communication builds trust, encourages collaboration, and helps others build on previous work. Visual aids such as graphs and infographics can make complex data more understandable. By communicating openly, researchers contribute to the advancement of science and its positive impact on society. Now, let's look at the role of education in promoting good data collection practices. Education and training are essential for developing strong data collection skills. Universities, workshops, and online courses provide opportunities for researchers to learn about new techniques and technologies. Ongoing training ensures that scientists can adapt to changing methods and maintain high standards. By investing in education, the scientific community supports the next generation of researchers and promotes excellence in data collection. Let's now discuss the importance of curiosity and creativity in biological research. Curiosity drives scientific discovery, and creativity leads to innovative data collection techniques. Biologists who ask new questions and think outside the box often make the most significant breakthroughs. Whether developing new tools, designing unique experiments, or finding novel ways to analyze data, creativity is at the heart of progress. Encouraging curiosity and creative thinking helps researchers overcome challenges and push the boundaries of knowledge. Finally, let's conclude our journey through data collection in biological research. We've covered a comprehensive overview of data collection techniques in biological research. From field observations to advanced laboratory methods, each technique plays a vital role in expanding our understanding of life. Accurate and ethical data collection is the foundation of reliable science, driving discoveries that benefit society and the environment. As technology advances and new challenges arise, researchers must continue to learn, adapt, and uphold the highest standard. Thank you for joining us on this journey through the fascinating world of biological data collection.